Hello guys, how are you all doing? It's been a while since I made a video. I've been, trying to catch up with a lot of things. I started a story about some animation, so it's been a lot of work lately, okay? So today I want to talk about my ulcerative colitis research, okay? So ulcerative colitis is an inflammatory bowel syndrome, is a chronic inflammatory bowel syndrome affecting the intestinal tract of the digestive system, okay? So the main cause of this disorder is not known yet, but research has shown that key bacteria in the gut, because the gut is kind of there's something called the microbiome. It's got beneficial bacteria and non-beneficial bacteria, okay? So when the non-beneficial bacteria are in a high amount or in copious amount, there is something called gut dysbiosis. It also leads to dysfunctional peristalsis of the gut. So that's when inflammation occurs, and then diseases like ulcerative colitis, Crohn's disease, what's the one called diverticulosis, they all occur together, okay? So there's just key bacteria in the gut that is causing it, okay? And notable few bacteria that has played a key role in the development of ulcerative colitis are invasive E. coli, which releases sugar-like toxins with its PKS genes, and the other one is Clostridium difficile. It releases toxins using its TCA and TCB genes, okay? So I'll talk about the symptoms of ulcerative colitis. So symptoms include gut spasms, bloody stool, unusual weight loss, things like that, okay? So those are the symptoms, okay? And then my research is basically using so there's this thing called the gene knockout system. There are different types. There's zinc finger. There's also this one called suicide plasmid vector, and there's CRISPR-Cas9, okay? But my main focus is using the CRISPR-Cas9. So this gene knockout system helps in gene deletion and gene sequencing deletions and gene sequencing additions, okay? So we can take away sequence and replace with a better sequence, okay? So what we're trying to do is we're trying to use beneficial bacteria genes like lactobacilli and bifidobacterium. So these are beneficial bacteria in the microbiome. So we're trying to use their sequences and replace with the sequences of the non-beneficial bacteria, okay? Because if we can do this will help yield or promote something called